Hello everyone, and welcome to Lab Manager's Product Spotlight webinar series. These webinars are designed to help attendees understand a specific proprietary service or technology and where and how it can be applied in an effort to help labs operate more efficiently. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's webinar entitled From Wells to Westerns, Investigating Cellular Pathways on a Single Platform. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the presentation. Many of you know the drill, but for those of you joining us for the first time, you can simply type your query in the question box located at the bottom left-hand side of your screen. We'll try to address as many questions as possible during the Q&A period. However, if we happen to run out of time, I will forward any unanswered questions to the presenter, and they can respond to you via email. Additional resources can be accessed through the resource widget located at the bottom of your screen. You may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. At this time, you may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website. At the end of this webinar, we'll share that link with you, as well as the contact information for our speaker. At this time, I'd like to introduce our speaker today, who joins us from Molecular Devices. Dr. Cathy Olson has been an application scientist at Molecular Devices since 2004, where she is responsible for developing a broad range of applications for microplate-based detection systems, including cell-based, biochemical, and imaging applications. Cathy received her PhD in Cell and Developmental Biology from the University of California, Davis. Her thesis work on the role of forkhead genes in acidian development was published in several key developmental biology journals. Cathy's postdoctoral research focused on how transformation affects normal growth processes of human mammary epithelial cells, as well as the role of hedgehog interacting protein in hedgehog pathway cell signaling. Thank you for joining us today, Cathy.
Hey, thanks, Trevor. So let's get started. I would like to spend about the next half hour talking to you all about a topic that I like to call From Wells to Westerns. We'll talk about investigating cellular pathways on a single platform. So I'll be describing to you how we can use a single instrument and a set of reagents to investigate our biology with one complete system. So we'll be looking at a number of different assays that are performed on the same platform, including measuring protein activity and expression, monitoring cell counts and cell confluence, and also quantifying cell viability. So the system that I'll talk about is called the SpectraMax i3 multimode detection platform. So it's a multimode plate reader with 3 detection modes automatically on board. Those are absorbance, fluorescence, and luminescence. And in addition to these onboard modes, it has cartridge-based expandability. So several cartridges, a few of which I'm showing here, can be added to the system to add additional detection modes to the system. So among those are Western blot detection, which I'll be showing you today, TR-FRET cartridges, including HTRF for screening, alpha screen, and coming soon, a nano TRF cartridge. We also have the capability of adding imaging to the platform, and that's through the MiniMax Imaging Cytometer, and I'll have a few more details on that in just a moment. That includes imaging in transmitted light channels. That includes StainFree technology for cell counting without dyes, and fluorescence imaging. We also offer, optimized on the platform, a number of assay kits, and these include the Early Tox Cell Integrity Kit, a cell viability assay that I'll be addressing today, Early Tox Cardiotoxicity Kit, and the ScanLater reagent kits for Western blots, which I will also discuss today. So to start off giving you a few more details on the SpectraMax MiniMax Imaging Cytometer, this is an additional capability that can be added to the instrument at any time by the user in their lab, and it's shown here as the orange part on the base of the plate reader. So again, that can be added at any time and adds imaging capability to the plate reader system. So it allows users to combine cellular imaging and microplate assays into a single workflow. It enables you to count cells and also measure cell confluence without dyes using our patent-pending StainFree technology. And it also allows you to expand your cell viability and toxicity applications with multi-channel fluorescence imaging functionality. And all of these functions are run through the SoftMax Pro software, which performs acquisition of images and also image analysis very quickly and easily on the system. A few more details on the ScanLater Western Blot cartridge. So this is another detection cartridge that can be added by the user at any time to the base system. So it's comprised of a number of different components. So the first is the cartridge itself, and the other is a membrane holder, which is shown on kind of the bottom middle of your slide. That's an SPS footprint tray that holds the Western Blot in place during reading of the membrane on our system. We also offer a number of reagents and assay kits for the Western blots, and these include europium-labeled secondary antibodies. And the detection and quantitation is fully controlled also by SoftMax Pro software. So everything I'm going to be showing you today, data acquisition, image acquisition, and all analysis and graphing will have been done in SoftMax Pro, with the exception of a few of the bar graphs. So the workflow for the Western Blot cartridge is basically the same as what you're used to if you're already doing Western blots. So on the right-hand side, I've outlined this workflow. So step one would be to apply samples to a gel and perform electrophoresis to separate the bands based on size. And step two would be to transfer to the appropriate membrane, and we have a number of recommendations for membranes that work particularly well on our ScanLater system that have low fluorescence background. Step three would be to block, and you can use your own blocking solution, or we also recommend using our ScanLater blocking buffer, which is provided in the kit. Step four, incubate with your own primary antibody of interest. Next step would be to wash, and again, we offer a wash buffer that works really well with the system, or you have the option to use your own. Step six is to incubate with our ScanLater secondary antibodies. So again, these are europium-labeled antibodies. We have several available now, including goat anti-rabbit, goat anti-mouse, donkey anti-rat, donkey anti-goat, plus a streptavidin-labeled europium if you have biotin-labeled proteins of interest. So there's a final wash step, and then the very last step is the step where we differ from all the other detection systems that are out there for Western blot. And in this step, you simply take your blot, put it into the blot holder, put it into the i3, and do a scan. And the nice thing about this is that it's not, because it's europium-labeled secondary antibodies rather than an enzymatic luminescent reaction, you have all the time you need to read your blot. You could read it immediately, or you can even let the blot dry out and read it weeks or even months later. It really doesn't matter. It's very stable, and the signal will remain strong for as long as you want to keep the blot. So I've given you a little background on the i3 system, the MiniMax Imaging Cytometer, and the ScanLater Western Blot cartridge. And now I'd like to jump into the biology. So we're going to be examining some aspects of the cellular response to heat shock. So there's a number of pathways that have been studied with regard to heat shock, and I'm showing you a pathway that was published in Journal of Applied Physiology. To give you an example, so in this example, heat shock activates kinase pathway, which activates several additional downstream elements, culminating in expression of caspase-3 enzyme, which then activates apoptosis in the cells. So there's a kind of a parallel pathway running here. When the Jun kinase at the beginning of the pathway is activated, that can also lead to induction of HSP70. Now, HSP70 is the heat shock protein, and here I just want to stop for a moment and just let you know that sometimes you'll hear HSP70 referred to as HSP72. There are different isoforms of this out there, including HSP73 and then HSP72, which is inducible. So all of these things, when you read the literature about heat shock, are all referring to what I'm going to be calling HSP70 today. So anyway, that gets induced, and that actually has been shown in some cases to have a protective effect on the cells and to prevent them from undergoing apoptosis. So we're going to show you a number of experiments that we've done today involving heat shocking of CHO cells, and we're going to look at some of the responses to heat shock in the following ways. So first of all, we monitor the morphological changes, and we do that by looking at the cells on the MiniMax system. And just for starters, we use the transmitted light channel, and we use our StainFree technology to get cell counts. And the second thing that we'll look at will be cell viability assays and apoptosis assays. So those are all done using the fluorescence imaging capability of the MiniMax system. And then finally, I'll show you how we use the ScanLater Western blot system to look at HSP70 protein induction in the heat-shocked and control cells. Okay. So I want to start off by giving you a little bit of background and information on just how cellular responses to heat shock are known to vary. And they can vary a lot depending on the cell type and even characteristics of a particular cell population, passage number, growth conditions, and all of those can affect the cellular responses when you heat shock them. So we had done some initial experiments in CHO cells, actually a number of experiments, and we found that under different growth conditions, the cells would experience more or less death in response to heat shock. So we determined that there are two conditions that we wanted to test and kind of compare and contrast as far as the responses that we're measuring for the heat shock. So we picked two growth conditions, and I'm going to call those today normal cells and stressed cells. So the normal cells were cells in which we changed the media very regularly. We tried to keep them as happy as possible. We passaged them at 80% confluence or less at all times to keep them from getting overcrowded. Now the stressed cells were cultured in separate flasks. They were allowed fewer media changes, so in effect they were serum starved, and we would allow them to go without a media change for about 3 days before we seeded them for assay. So they are serum starved. And they were also allowed to become overconfluent just prior to seeding our assay plates for the experiment. At this point, we could see that the stressed cells were in full contact with each other. The pH of the media was clearly higher than normal, and we also could observe some floating cells in the culture. So this is what we're calling stressed. Okay. So the workflow that we're subjecting these normal and stressed cells to is outlined in this slide. So first, for beginning, we see the normal and the stressed cells separately into 96 and 6-well plates. So the 96-well plates are the format that we're running our cell-based assays in and imaging. And the 6-well plates we seeded just to make sure that we had plenty of protein for the Western blots. That turned out to be way more protein than we needed and could actually be scaled down. So the 6-well is very generous plating. So we took half of these plates and kept them at 37 degrees as controls. And the other half of the plates were placed at 45 degrees Celsius incubator for 90 minutes to heat shock them. After the initial heat shock period, the cells were put back into a 37-degree incubator, and then they were allowed to recover for either 6 or 24 hours. And then at that point, we took the plates out at 6 or 24 hours and assayed them for proliferation, cell viability, and apoptosis. In the parallel samples, we collected and assayed cell extracts and ran the Western blot for the HSP70 induction. Okay. So the first bit of data that I want to show you is how we monitor the cell morphology. So on the left-hand side, I'm showing you two images of healthy cell populations. The top image are the control, which were maintained at 37 degrees, and the bottom are the heat-shocked cells, which were heat-shocked at 45 degrees for 90 minutes. On the right-hand side, I'm showing you two images of the stressed cell population. The top is the control, 37 degree, and the bottom are the heat-shocked at 45 for 90 minutes. So these were all imaged in the transmitted light channel of the MiniMax Imaging Cytometer. And right away, we could see by eye that there are some differences among the different treatments. So in the healthy control cells, we see what is normal proliferation. And again, this is after 6 hours after the heat shock. And in the heat-shocked healthy population, we may see a little bit less proliferation, but there's not a really marked difference between the control and the heat-shocked in those healthy cells. In the stressed cell population, we can see that the controls appear fairly normal, and then the heat-shocked population is already displaying some visible signs of rounding. There are more rounded cells with a sort of apoptotic morphology. And we can also see that there appears to have been less proliferation. You don't see as many clusters of cells in that bottom right image. And here the yellow arrow is pointing to a cell that has a rounded morphology that suggests apoptosis. So that's actually the first thing we wanted to measure is how many cells are there in the wells 6 hours after heat shock and 24 hours after heat shock. So we can do that initially without doing any staining or dyeing of the cells by using the MiniMax and the StainFree technology for cell counting. So on the left-hand panel, I'm showing you an image of some cells. These are the control population, just for reference. Again, these are CHO cells. And the top image is just showing the basic image before we do any analysis to it. And the bottom image is showing what happens when we pick a setting in the software to identify the cells in the image and then apply that setting to the images. Purple masks are showing where the software has identified cells in that image. So this can be done in different ways through the software. There are a number of predefined settings that we have where you can simply click on a setting and have the software automatically find all the cells in all of your images. If you have, and that works for a lot of cells that have common morphologies for which we've developed the settings. If you have cells that may be a little different, different shapes and morphologies and things than we've tested in-house, you have the option to do a supervised machine learning, as we call it. And in that, you actually draw on the cells in the image using your mouse, and you indicate where the cells are, and then you indicate where the background is. And you can essentially teach the software to recognize your own cells of interest. And that works really well for a wide variety of cells. Okay, so that is the kind of analysis. We actually used a predefined setting that works really well for CHO cells in the analysis that I'm showing you here. So I've got a couple of graphs showing you the cell counts that we came up with for the control and heat shock cells. And I'm showing you in each graph the normal and the stressed cell populations. All the blue bars represent control cells that were grown at 37, and the orange bars will represent cells that were heat shocked at 45 degrees for 90 minutes. So you can see even after 6 hours, there appears to be a slight reduction in the number of cells present, even in the normal population, when you heat shock them. And that difference is more pronounced in the stressed population, even at 6 hours. So you can see there are fewer cells overall, and when you heat shock them, there's even a more reduced number of cells present. So now by 24 hours, we can see that there's definitely a lower rate of proliferation in those stressed cell populations that were heat shocked. You can see that the cell numbers actually do go up in the normal and the control populations, but the stress cells that were heat shocked remain very low. So we can already begin to quantify the kind of responses that are happening in terms of cell counts and proliferation rates. So the next thing that we wanted to do is to see if the cells are dying, and if so, by what mechanism. So the first assay that we did was an apoptosis assay. And for this, we used the cell event caspase 3,7 green detection reagent. It's a fluorescence imaging assay that allows us to count individual apoptotic cells. So it works on the principle of a substrate, which is a fluorogenic DNA dye with a DEVD substrate moiety that gets cleaved by the caspase if it's present. That substrate crosses the plasma membrane of the cell where it gets cleaved by the caspase, assuming the caspase is active, to release a high-affinity DNA dye. Now that dye, once it's freed, can migrate into the nucleus and it stains DNA there bright fluorescent green. So it is possible to do the assay purely on live cells. They can also be fixed in formaldehyde after assay to do additional tests on them. Now the data that I'll show you today was done on all live cells, so it's not necessary to fix them. We imaged them in the green fluorescence channel of the MiniMax, and then we use SoftMax Pro to count the apoptotic cells. So we set up the software to define a threshold of size and fluorescence intensity so that the software would automatically identify all of the apoptotic cells in all of our images. And the imaging setup and the analysis really takes less than 5 minutes. So here's our response. So again, we counted the green fluorescent or apoptotic cells, and I'm showing an image on the far left of the yellow arrow indicating an apoptotic cell, which is very bright green fluorescent. And you'll also see nearby a number of flatter, more normal-looking cells with a normal morphology, and then some kind of shades of green in between. So when the cells are beginning to express the caspase, they may be a little bit less bright green. But we can set our threshold to identify the ones that are clearly already apoptotic. So what I've done here with the data is to have the software count the number of apoptotic cells and then normalize that to the StainFree cell counts that I showed you in the previous slides. And that way I could obtain the percent of apoptotic cells present in each and every well. So what you can see, looking at the graph below on the left, and again we're looking at percent apoptotic cells on the y-axis versus the conditions that the cells were grown under on the x-axis. And the first graph is for after 6 hours post-heat shock. So you can see in the normal cell population, which is the bars on the left, there's very little apoptosis going on either in the control or the heat shock condition. There may be a slight increase, but it's really not significant. In the stressed cell population, you do have a few more apoptotic cells in the population, even in the control cells, and then when you heat shock them, that number goes up even higher. So after 24 hours, we see a much more pronounced difference. So here, the normal cells have also experienced an increase in the percent apoptotic cells in the heat shocked group, and then the stressed cells have a much, much higher percentage apoptotic cells after 24 hours. So whatever we've set in motion with our heat shock as far as the pathways is continuing to produce apoptotic response in the cells after 24 hours. So next we wanted to know if the cells were dying purely through apoptosis or if we could get some clues as to whether they might be dying through other mechanisms. So we wanted to run a cell viability assay. And the one that we chose to run is the Early Tox Cell Integrity Kit from Molecular Devices. So this is a kit that works on the principle of identifying live cells and dead cells through the use of 2 fluorescent dyes. So the first dye is called live red dye, and that's a cell permeant dye that's able to cross intact membranes of live cells. It can also go into dead cells. And it goes to the nucleus and binds to the DNA where it becomes highly fluorescent. So each and every cell in the population, whether it's live or dead, will be stained bright red fluorescent. So the second dye we call dead green dye, and that's because it is a membrane impermeant dye. It's only able to get into cells whose membranes are already compromised or whose cell integrity is already compromised. So that the green dye goes into the dead or dying cells and also enters the nucleus and then binds the DNA there. So every dead cell in the population will be labeled red and green. So that's how we tell those cells apart. Live cells are red, dead cells are red and green. And we can have the software, SoftMax Pro, identify which cells are live and dead through our image analysis. And that's what I'm showing you here. So the way that we quantify cell viability is that we have a couple of reference images that are acquired initially when we start the reading. And those images are used to essentially train the software what's live and what's dead. Now the first step is to have the software identify all of the cells through the red channel, so it finds everything that's a red nucleus. So the second step is to further classify those cells as live and dead. And so we do that simply through a classification analysis where we just click with our mouse on a few representative samples of live cells and a few representative dead cells. And from those few clicks, the software is able to figure out which ones are live and dead in the entire image. So once we set up the initial analysis, which only takes a minute or two, the software will automatically identify all the live and dead cells in all of the images. And the entire thing takes only a few minutes. So the images that I'm showing you here on the top are just the raw images with the red cells in a mostly live population on the left. And you see a few dead cells in there, but mostly live. And then on the right is a mostly dead population that was treated with a compound that causes them to become apoptotic. So you see almost everything in there is green. And the images on the bottom are the classified images from the software, where the software has applied red masks on the left-hand image to everything that's live and blue masks to everything that's dead. So you can see most everything is a red mask on the left, and most everything on the right has a blue mask because it's dead. Okay, so we applied this kind of analysis to the heat shock samples. And I'm showing you on the left-hand panel an image that's just an example of some heat shock cells, and we're showing some live ones and some dead ones. The dead one has a green arrow indicating it, and the live one has a red arrow next to it. So we can see that heat shock does indeed alter cell viability in general. So we have an increase in cell death that occurs after heat shock. And you can see that even at 6 hours. So what are we looking at here? We have graphs showing the percent dead cell on the y-axis, again, versus the growth conditions on the x. So percent dead cells in the normal population do not increase very much with heat shock. So it's a fairly low percentage of dead cells overall. In the stressed population, we do see an increased number or percentage of dead cells in the population, and that does increase a little bit with heat shock. So this is really reminiscent of what we're seeing with the apoptosis results. And that's echoed in the 24-hour results, where we see an enhancement of the percent dead cells in the heat shock treatment. So again, what this suggests is that all or most of the cell death that we're seeing is probably due to apoptosis, and we could certainly take this hypothesis and further investigate it through some follow-up assays on the same system, for example, using things like caspase inhibitors. But this initial data does give us the hypothesis to work off of. So what we've learned so far from these imaging assays, again, is that the percent apoptotic and the percent dead cells overall observed with the two imaging assays were fairly similar. And so from that, again, we're hypothesizing that all or most of the cell death that we're seeing in response to the heat shock is probably due to apoptosis. And we've also seen that inducing stress via serum starvation, overcrowding, et cetera, in cells prior to the experiment did make them quite a bit more susceptible to the heat shock-induced apoptosis. Okay. So since we also know from the literature that the heat shock does induce HSP70, and that protein has known to have a protective effect on cells from apoptosis, we next looked at the level of the HSP70 protein that's induced in our experimental cells. So I first wanted to talk a little bit about the Western blot sample prep, because that's another thing that the SpectraMax i3 can assist with. So again, from our 6-fold plates, we had collected and lysed some control and heat shock cell samples, ran a BCA protein assay on the SpectraMax i3 reader using its absorbance detection mode, and we were able to calculate the concentrations automatically with SoftMax Pro. There is a pre-configured protocol in the software that allows you to set up the standard curves for the BCA automatically, and from those standard curves, the unknowns or your sample concentrations are automatically calculated. So it simplifies this step greatly. So once we have our quantitated protein samples, we can determine how much to load on each gel. We settled on 3 micrograms. That looked good for each lane on the gel. So we ran the electrophoresis, blotted the gel onto a BioRad immune blot low fluorescence PVDF membrane. And that one is one of the recommended membranes for use with the ScanLater system because it has a very low fluorescence background, and it enhances our signal to background and gives us better results. So after we blotted onto the membrane, we ran blocking steps, primary antibody, and the primary antibody here was the rabbit polyclonal antibody against HSP70. Did a series of wash steps. We applied our secondary antibody, which in this case is our ScanLater European labeled goat anti-rabbit secondary, and then a final set of wash steps. And at this point, we have the option to either put the blot immediately in the instrument and scan it, or we could do it the next day. We could even let the blot dry out in a drawer in the lab and read it weeks later. So it's very flexible at this point. So we read it. We analyzed the band intensity through the SoftMax Pro software. And that's a whole automated analysis that I won't have time to get into in great detail today, but we can point you towards some additional resources that have more information on how that's done. Okay. So here are the results condensed down to the panel on the left. So the panel on top is the normal cell population, on the left is control, and on the right is the heat shock lane. And the bottom panel are stressed cell population, on the left control, and on the right heat shock. And I also have a graph, so we were able to do quantitation of these bands. We can see that they're clearly induced, especially in the normal cells, but we can actually quantitate this induction using the ScanLater quantitation and SoftMax Pro software. So on the y-axis, I'm showing you normalized HSP70, which normalized HSP70 in this case means integrated density minus the background on the blot expressed in millions. So in the normal population, we see a 48-fold induction of HSP70 in response to heat shock. whereas in the stressed cells, we only see about a 1.5-fold increase when they're heat shocked, and the overall level of expression of HSP70 is quite a bit lower than in the normal cells. So the HSP70 in the stress cells never even reached the high levels that were in healthy cells. And we feel that as a result, the stress cells may have had less protection from apoptosis, leading to the higher numbers of apoptotic cell counts that we observed. So that's one explanation for the results that we're seeing in this case. Okay. So the conclusions that we're drawing, again, are that higher initial expression of the HSP70 in the stressed CHO cells would correlate with an enhanced response to heat shock compared to the normal cells. We also saw that the percentage of apoptotic versus dead cells was pretty similar under heat shock conditions, and that's suggesting the mechanism for what's happening to the cells may be primarily through apoptosis rather than necrosis or other death pathways, but those can be further investigated on our i3 system. Okay. So just to recap everything that I've shown you today, we've looked at cellular responses to heat shock, and we've looked at a number of different aspects of the pathway, including cell viability, apoptosis, protein induction, and also morphological changes. So we studied those morphological changes initially using the StainFree technology, where we were able to count cells without adding dyes. We looked at our cell viability using the Early Tox Cell Integrity Kit, where we could identify live and dead cells and determine the percent dead in each population. We did a fluorescence imaging cast-based assay to get into more detail on the mechanism of the cell death and saw that most likely this was occurring through apoptosis pathway. And then finally, we used the ScanLater Western blot system to look at induction of HSP70 in the control and experimental populations and see that we get less induction in the stressed cells, potentially causing them to be more susceptible to apoptosis. And all of this was done on our complete solution, the SpectraMax i3 with the MiniMax and the ScanLater Western blot detection system. So I hope I've given you some ideas for how you could use this system in your own lab. Okay. And finally, I want to thank everybody for attending, and I also want to thank my colleagues, Huang Ha, who did almost all of the cell-based expression or cell-based experiments that I'm showing you today. He did the heat shock experiments and also collected the protein samples and ran the protein assay. And also Vanita, who did the Western blots, and she's our Western blot guru here. She was very helpful in helping to prepare the samples to run the blots and help us with data analysis there. And then Celeste Glazer is our product manager for the i3, and she's provided some of the great slides that I showed you at the beginning of the presentation. Okay. And before I close and or open up for questions, I wanted to invite you to visit us at one of our new features on our website called At the Cell Counter, and I've got a link included here. This is a monthly feature that will show you how to use the SpectraMax MiniMax system to do cell counts on a variety of cell types. So we have tips and tricks from application scientists, including me, and we'll have a new cell type featured every month. This month it's rat aortic endothelial cells, but it's something new every month for you to see how our system can be used for your cell type. And also, I think on your screen displayed on the left-hand side, we've included a few resources, some application highlights that you can go and check out to find out more detail about the assays and the detection systems that I've talked about today. So, at this point, I would like to go ahead and open up for questions.
Well, thank you very much, Cathy, and we will go directly into our question and answer session. We have a number of questions that have come in from the audience, and for those of you who may have joined us late, you can ask questions of Cathy simply by typing your query into the question box, which is located at the bottom left-hand side of your screen. So, Cathy, how soon would you need to read your Western blot after adding the europium-labeled antibodies?
So the blot, as I mentioned, it can be read anytime from weeks to months after it's initially probed with secondary antibody. And we've kept some blots around for several months now, and we take them out periodically and put them on the system and read them, and they still look great. So they could definitely be dried and saved and reread as much as months later. So it's very stable.
Great, thank you. What is the sensitivity of the ScanLater Western blots?
Yeah, so we have done extensive studies on the sensitivity, and I believe we've demonstrated that about 2 picograms of a purified GST protein can be detected on the ScanLater Western blots. Of course, that number is going to vary, according to the protein that's being analyzed, but we're definitely seeing in the single-digit picogram levels of sensitivity. So that's very sensitive, and it's favorably comparable. It's at least as good, if not better, than a lot of the chemiluminescent systems that are out there.
Great, thank you. And Cathy, does the MiniMax automatically detect and count cells?
Yeah, it can definitely do that. So there's a number of ways. So detecting the cells, it can do through any of the 3 channels. So it can do it through transmitted light channel using the StainFree analysis. And there, as I had mentioned in the presentation, for things like CHO cells where there's a common morphology, we have a lot of predefined settings that can be applied, and those are available for several different common cell types, including some fibroblasts and some suspension cells. And if a user has, a more strange-looking or different type of cell that's not got a pre-configured setting, they can definitely train the software to recognize those cells of interest. And yeah, we've had a number of users try their cells on the system, and they were really skeptical initially, but then they discovered that, in fact, the system could be easily trained to recognize even their strange-looking cells. So it can work really well. So I hope that answered the question. For other detection besides StainFree, we have the fluorescence imaging, and there are also pre-configured settings in there for nuclei or for cells, and those can simply be used kind of a one-click analysis of cell numbers and fluorescence channels.
Great, thank you. Now, can it be can it work for isolated organelles such as mitochondria?
Yeah, so we do. I don't think it's listed on the resource page this time around, but we do have an application note looking at mitochondrial function, and that was analyzed on the MiniMax. So we used a mitotracker dye, and we were definitely able to see differences in mitochondrial staining under compound treatment conditions. So yeah, those can definitely be imaged as well.
Great, thank you. Now, you loaded 3 micrograms of protein. Is that a default amount, or can it go up?
That it can go up. So we settled on that amount because that's what gave us the best image or the best data for the particular protein that we were looking at. So if you have something that is expressed at lower levels, then you could definitely increase the amount of lysate that is loaded. So I know in-house we've loaded quite a range of volumes of lysate, and if the questioner wants to get in touch with me offline, they can definitely do so, and we can talk about it further, and I could try to find out if there's limits. But I know it's very versatile in terms of how much you can load. It just really depends on the target that you're looking for.
Great, thank you. Now, for people who are looking at investing in new systems, what are the main considerations they should be looking at?
So I think a really important thing, especially now when research is moving so fast, is to be sure that you have a system that's going to be versatile and that's going to be flexible enough to allow you to do what you're going to need to do, not only today but in the years to come. And so, a system where you can add additional detection modes if your assays change, or if you can add the Western blot detection and the imaging, if you start to get into some of those areas, would be, I think, an ideal system. And the fact that the i3 already has 3 modes on board that a huge number of plate reader users are already using. So we're already doing a lot of absorbance, fluorescence, luminescence assays. But these assays may give them ideas on things that they want to do that they would rather image. And so it's a very versatile system. And I would think that future expandability would be a very important feature to think about, as well as sensitivity and things like that.
Absolutely, absolutely. Well, Cathy, I'd like to thank you for providing an excellent discussion today. And this brings us to the end of today's webinar. If you have any further questions, please consider reaching out to Cathy directly. Her contact information, as you can see, is available on the screen now. Just a reminder that today's webinar was recorded, and you will receive an email notifying you when this on-demand video is available. On behalf of Lab Manager, I'd like to thank Dr. Olson for joining us today and Molecular Devices for sponsoring this Product Spotlight webinar. This concludes our Product Spotlight. Oh, thank you. And I'd like to thank all our attendees for taking time out of their busy schedules to join us today. You may wish to join us for our upcoming Tech Trends webinar, which discusses recent trends in laboratory automation. This webinar is occurring tomorrow, Thursday, February 26th, from noon until 1:30 p.m. Eastern Time. Please visit www.labmanager.com for details. Thank you for joining us today, and have a great day.